So, I'm surprised Gracie's not here.
She's on the IUK visit too.
I know, but I'm surprised she.
Yeah, I'm recording for them.
Oh, is that it?
Yeah.
Okay. Chapter 18. All right, we'll kind of get you into this and then go from there. Okay. What was I gonna say? What was chapter 18 called? It contains operands. So 18, it contains repressors, right? Any? It deals with prokaryotes, but it also deals with eukaryotes.
Gene expression.
What's that?
Gene expression.
Yes. Yeah, so 18 is the gene regulation chapter. Okay, so the first part, Christian, you guys are talking to me. There's prokaryotic gene regulation, which deals with operons, and then the rest deals with eukaryotic gene regulation. But the most important part is the prokaryotic, okay? Okay, so first of all, repressible operons. Okay, what's the example of a repressible? Trip or lac? Sure. Yeah, this is the trip. Inducible is the lac. Okay. So let's do repressible. Okay, repressible operons are always on or off? On. Okay, they are always on. Okay, so I'm going to draw the operon. The most important part that really matters is this region called the
Active site.
So it used to be you dial 0 on the phone, you would get the
Operator.
Yeah, does that still exist? I've never dialed for the operator. Yeah, I just. Okay, so there's the operator. Okay, do you notice the other Lego piece isn't on it? Okay, so if I go over here and I draw the other Lego piece, okay, what is that Lego piece called? Yeah, this is the repressor protein. Okay, and since the repressor protein's not on there, this is switched, it's on, right? That's why it's always on. So, got that? That's like part 1, right? So now we're making a bunch of trip, right? And now we have too much of it, okay? And we need to do what to the pathway? We need to turn it off, right? So what is trip going to do? Yeah, it's gonna bind to the repressor protein. Repressor protein is going to go change shape, so then it will fit in the operator, okay? And when that happens, now RNA polymerase can't do what? You guys looking? RNA polymerase can't go through, right? So that's how you would shut it off. Okay, so the presence of tryptophan does what to the operon? What's the word? Represses it. That's why it's called repressible. Is it back in your brain now? You got it? Okay, now let's do inducible. Can I clear it? Yes. It's, you guys ready for the weekend?
Yes.
All right, so we'll do this. Okay, there's my operator. Okay, this is the lac operon. Okay, the lac operon is always off. Okay, so I need to turn this off. right? So I'm going to put a little repressor protein here, okay? Now if you notice, there's no lac in that repressor protein, okay? So now, the bacteria runs into some food, and it wants to eat it, okay? It runs into some lactose, okay? The presence of lactose, the lactose binds to the what? Repressor protein, making the repressor protein come off, okay? And when that happens, this whole thing is turned on because the presence of lactose does what? What kind of operon is this? It induces it. It turns it on. It makes it happen. Okay? So you see how the repressible and inducible, they're opposite of each other? Okay. That's it. You got it? It's not that hard, is it? I know I'm a teacher. When Mr. Bayamita was here, he would just say that's 5th grade math. You guys ever experienced the 5th grade math thing? Okay, so can I move on? All right, moving on. All right, the rest is eukaryotic gene regulation. So there's like 7 different things. These are the ones that I put on the test, okay? So if you know these, you know the eukaryotic. Okay? So here it is. If you methylate something, you're giving it a methyl group, okay? Methyl group is binding to DNA. What's it doing to it? Turning it on or turning it off? Meth is bad, but off, right? So methylation turns genes off, okay? Histone acetylation, okay? So you remember this? The beads on a string, the histones, the red things are histones, the orange things DNA. Okay, why does DNA bind to the red histones? But why is it attracted to the histones? Yeah, remember DNA is negatively charged, and these histones have what charge? Positive. So that's why they're able to like stick to it, right? But if it's tightly wound, RNA polymerase can't get in there and read it, okay? But if it's loosely wound, then that means what can happen? Yeah, RNA polymerase can come in. So when you acetylate something, you're making it more, it's loosely wound, which does what to the gene? Turns it on. So methylation off, acetylation on. Okay? All right, heterochromatin and euchromatin. Which one is the tightly wound DNA that's off, and which one is the loosely wound DNA that's on? And I'm going to give you a little device to remember this, but do you know the answer? Hetero is off, so it's tit. Oops. Off. Yeah, tight, G-H-T-L-Y. It's tightly wound. Okay, so it's off. Euchromatin is loose, so it's on. So if you remember the notes, here's the nucleus, right? So here in the nucleus is a bunch of DNA. This region is really dark, and this region looks like that. You see what I mean by tightly wound and loosely wound? This is dark, that's heterochromatin. This is light, this is euchromatin. Okay. Another way to remember this, a little weird device, you use euchromatin. Get it? Meaning you're going to need it to make proteins, it's on. You use euchromatin. All right, and then the last one is siRNA. So siRNA tag, so here is RNA. Okay, RNA, siRNA, right here, tags RNA. Remember my analogy with the Star of David, Nazis, Holocaust? Yeah, if you've been tagged, what's gonna happen to you? You're basically you're gonna go to concentration camp, right? If RNA has been tagged, what's gonna happen to it? It's gonna be all chopped up and not used, okay? So siRNA tags the RNA to be basically eliminated. So if it's gonna be eliminated, it's turning it off, of course, right? Okay, you got it? That's chapter 18. So, okay, 19. All right, 19 is the virus one, okay? Okay, so bacteriophage, right? What's inside of this? The DNA, okay? Now, the outside coat is made of protein. What's the word? Capsid. It's a protein coat. Hey, by the way, who are the two people that prove that they inject DNA into a cell? Hershey and Chase, right. Because they wanted to see, does the capsid go in or does the DNA go in? Okay, here's another virus. All right, so now this is called an envelope virus. Okay, an envelope is made of a membrane, like a cell membrane. Okay, this is the capsid, and then of course there's your genetic info. So envelope viruses, all viruses have a capsid. It's just some viruses have an envelope, some don't. Okay, so HIV is a good example of an envelope virus, right? Because on the surface of this are little receptor proteins, right? And if this is your body cells, you have these receptor proteins on your cells. What's this virus going to be able to do? It's going to dock, its receptors are going to dock to your receptors. This membrane is going to fuse with your membrane, because membranes are membranes, and then it's going to put all its stuff inside you. Okay? Good times. Right. Okay. So that's capsid envelope. So glycoproteins, see if you remember anything from earlier in the year. I drew glycoproteins, where are they?
They're on the base.
Yeah, these things are the glycoproteins, right? The receptors. Yeah, those are
I
glycoproteins.
thought
Well, these are proteins, these are proteins. They're both proteins. They're both actually glycoproteins. Yep. Okay, so next one, lytic and lysogenic. These are easy. Lytic is just another way of saying lysis. And lysis means to break apart. So a lytic viral infection, the virus infects you and quickly makes more viral particles, and then what happens to your cell? Yeah. And what happens over several days? How do you start to feel? Sick. And then you might miss like 2 weeks of school, something like that. My Christian jab, it's my Christian jab. So, and then we have lysogenic. Lysogenic, the virus infects you, but what does the viral DNA do? It stays and becomes part of your nuclear DNA. It fuses with your nucleus, the DNA in your nucleus, and then it goes dormant, okay? And then certain things can make it show up again down the road. There are certain STDs. You guys ever learn about STDs in health? Okay. There's certain viral STDs, once you get them, you have them the rest of your life. Yeah, you have to deal with it. But what's the one I talked about? Cold sores, herpes, right. Herpes, the non-STD type, is a lysogenic infection. You're stuck with it forever. So you can take something to suppress it if you want, or you just live with it, okay? That's the difference in lytic and lysogenic. So it goes dormant, and then maybe stress, maybe final exams are coming up, you suddenly, a cold sore shows up, right? So it leaves the lysogenic cycle and enters the lytic cycle. How do you know? Because you see, like I said, the herpes is a great visual of the virus expressing itself. Okay? All right, there's the difference. All right, retroviruses. So, HIV. Okay? You don't have to know this, but human immunodeficiency virus causes what disease? AIDS, right? Takes about 10 years-ish before it shows up. But it's a retrovirus, okay? So retrovirus, retro means backwards in this case, right? So what do I mean by retro? Yeah, so what's its genetic info? It's an RNA virus. COVID is an RNA virus, okay? So here is, now we're going to say this is RNA, okay? Inside, so this is exactly what HIV would look like. It's an envelope virus with a capsid and it has its RNA, but it also puts some enzymes in your cell when it infects you. What are those enzymes called? Reverse transcriptase, okay? So reverse transcriptase, so here's the RNA, okay? Here is reverse transcriptase. Reverse transcriptase is going to take RNA and convert it into DNA. Remember reverse transcription, right? And that DNA is going to become part of your cells, and now it can express itself forever if it wants to, okay? So that's what reverse transcriptase is. And because there's no proofreading, when you convert RNA to DNA, it always mutates. That's why retroviruses are hard to come up with vaccines for, because they're constantly mutating, okay? So they have a very high mutation rate. No proofreading, okay? Okay, you good on that one? All right, vaccines. What's a vaccine? So usually a vaccine is a weakened or dead form of the virus injected in your body, okay? I mean, I could talk about RNA vaccines, but that's too recent to really put in here. But so smallpox, right? There's an outbreak of smallpox in Texas. Have you heard about that? Okay. Why would there be an outbreak of something that's pretty much been wiped out? Because there's some anti-vaccine people, and that's how it spreads. You have a group of people who don't want to be vaccinated, it's going to spread. So that's why it's back in Texas. But you won't get it as long as you've been vaccinated. So, yeah. But that's all vaccination is, just a weakened or dead form injected in your body. So. Okay, antiviral drugs. Okay, drugs that block the virus. That's all. So here's a good example. Let's go back to HIV. Okay, if there's a COVID antiviral drug, you can block COVID, but they only give it to people that are high risk, like people who have asthma and stuff like that. So what if, here's what an antiviral drug could do. This is one example. So here's your glycoproteins, your receptors. What if that antiviral drug blocked these receptors? Virus can't get in. Now it's going to be in your bloodstream a lot longer, that gives your antibodies time to fight it off and get rid of it. You never get it. Okay? And that's what an antiviral drug is. Pretty cool. Look how smart someone has to be to figure that out. Yeah, pretty smart. Okay, and yeah, this is what that's this stuff, right? The lytic and lysogenic, right? The virus puts its genetic info in the cell. That genetic info makes proteins. The virus reassembles and leaves. If it makes the cell burst, it's a lytic cycle. That's all. Okay. All right. Any questions on that one? Last chapter. Can I clear it? Yeah. Okay. So what was chapter 20 about?
Like plasmids.
Plasmids and electrophoresis.
Fingerprinting.
Yeah, fingerprint, right? So this is the DNA technology chapter, okay? How do you insert a gene into a plasmid? Remember that lab we did? Yeah. So think of what we did. You took
You
the,
denature it.
that you're getting the PCR. The lab where we made our bacteria glow. You're in electrophoresis.
AMP.
You had to, this is the bacteria thing. Yeah, so we used heat shock, okay? And what heat shock means is you had it on ice, and then you had it on heat, and then you put it back in ice, right? Because if here's your bacteria, and here are these little plasmids, we got to get these guys to go in, right? So you had it on ice, you put it on in the hot water bath, and you put it back on ice, and if all went well, and everyone got it to work this year, okay? I had my doubts, prove me wrong, that's good. You got them to go in, and how did you know? You had glowing bacteria. And other bacteria just never grew, well, some bacteria grew in the presence of ampicillin and things like that, okay? So, going with that, you had 4 Petri dishes, okay? So you had an LB plate, you had an LB amp, another LB amp, and then LB amp and ARA. Okay. So then this was negative, negative, positive, positive. Okay. So what does negative and positive mean?
Has that, or has the proteins.
Kind of. Has the plasmids, right. So positive means it has the plasmid, negative means it doesn't. So did bacteria grow in this petri dish?
Yes.
Yeah, because is there anything that's going to stop it? Okay? Growth. You had growth, right? Was there growth in this one? No. Why? There's no plasmid, right? You gotta have the plasmid that gives the bacteria the what gene? Ampicillin resistance gene. And one more time, what's ampicillin? It's an antibiotic, right? So there's no growth because there's no plasmid, it's not gonna work. Was there growth in the LB amp? Yeah, because the plasmid was there, so we had growth. Okay? Was there growth in the LB amp ARA? Yes. So we're gonna say grow and it glows. It glowed because what was also there? The arabinose. And the arabinose turned on the operon, which expressed GFP proteins, which is just like the lac operon. If lactose is present, it turns the gene on. If arabinose is present, it turns the gene on, right? Okay? So you know you're gonna have at least 1 or 2 questions about that lab, because that was a big lab, okay? Any questions on that? All right, how do you make cDNA? What does the C in cDNA stand for?
Copy.
Copy DNA. So that means you're going to take RNA and go backwards and make it into DNA. What do we use to do that? I just talked about in the last chapter. Yes, you use reverse transcriptase, which comes from what kind of viruses? Retroviruses, because retroviruses are RNA viruses like HIV, the HIV. Okay, got that? Okay, PCR. If I were to say summarize in basic person terminology, like if you asked Miss Maxwell to explain PCR, what would be her explanation?
I don't know.
No feelings, no emotions, it's science. PCR, it's a DNA copy machine. There's normal person lingo, right? Because it's a way to take DNA and make a bunch of copies of the same thing.
Wouldn't know what PCR is.
I know. But you guys do. I hope.
I think there was a reason why we were
So PCR heats and cools DNA, right? And when it heats it, so here's the DNA, double-stranded. It heats it, and then it copies it. Okay? And then it heats it again and copies it. Okay? So it heats and cools. But the heating is like 90 degrees Celsius, almost boiling. That's too hot for normal DNA polymerase to make copies of DNA. What is the name of the enzyme that we took out of thermal bacteria?
It's hard to follow.
Yes, so TAC with a Q, polymerase. Okay? So that's just a recall question. If you know that one, you got it. Okay, so how are we able to do PCR when it heats the DNA so high? It uses TAC. You gonna say TAC or TOC? It's TAC. TOC sounds weird. You say plaza.
I say Tequila.
To what? You say plaza or plaza?
Plaza.
Oh.
Wait, it depends on if I'm saying like stores.
Yeah, like there's a bunch of like TJ Maxx and Petco, and that's a plaza. But some people say
I think
plaza.
I just say both.
You say both. Okay, DNA microarrays. Okay, that's the spot plate question. Okay, so here's a tray with a bunch of little divots in it. Okay, and you put a chemical in the tray, and if it binds to the DNA, meaning the DNA's been switched on, it'll turn a certain color, and it's a way to indicate if genes are switched on or off. So certain colors will tell you if the gene is on, or certain colors will show you that the gene is off. That is a microarray. Okay? All right, last two things. I need my space. So can I clear this? No? Okay. Then we're done. Then you can take it easy, take a nap.
I just do this.
All right, can I clear?
Yeah.
Okay. Restriction enzymes. Restriction enzymes restrict the DNA, meaning they cut the DNA at specific sites. Not to be confused with those things that remove the introns, spliceosomes, which are made of SNRPs, right? Restriction enzymes, remember palindromes? Radar, right? If this enzyme cuts here, okay, let's do the complementary sequences real quick. So this is GAATC. How is this restriction enzyme going to cut? Right? So, GAATTC that way, GAATTC that way, it's a palindrome, right? Backwards, right? So when you cut it, it makes these little overhanging ends, right?
That's not the same.
Huh?
You have to change one of the T's to an A.
Oh, I forgot my second T. Yeah, get it.
I will see it.
Okay, hold on. You made me think. There. Still, all right.
There are two
Where am I messing this up?
Switch one of the T's.
Yeah, so.
It's because there's five.
Switch one of the T's on the top to an A. Oh,
Make this one what?
Wait.
I don't know, man. I give up.
Okay,
Keep the
AG.
T in the middle.
AG right here,
Yeah.
and this one C. Okay, that was hard. Okay, so anyhow, you have a palindrome, right? And when you cut it, it makes these overhanging sticky ends. So if I want to cut a plasmid, okay, I'm going to cut it with a restriction enzyme. And then here is a human gene. Am I going to use a different restriction enzyme or the same restriction enzyme? The same, because what do you, the bases have to match. If I use a different restriction enzyme, I'm gonna have a different sequence of those overhanging bases. All right, so we cut DNA with restriction enzymes, okay? Then so after cutting it, you then take up that DNA in your micropipetter and you fill the wells. By the way, you guys had quality gels this year. I was really impressed. So, good job. And then you turn the electricity on, and the DNA is going to migrate to the
bottom.
positive end. This is the negative end, right? Because DNA migrates to positive because DNA has negative charge. And then the bands are going to separate. So let's say this one produces these three bands. Which band is the small band?
The bottom one.
Yeah, this is the small one. This is the largest one, right? Why? Yeah, they're smaller, they're lighter, they can squeeze through the gel. These are big, hefty DNA fragments, they can't get through, okay? So then we do this. So who committed the crime? So here is CS, suspect 1, suspect 2. Suspect 2. If it's a paternity case, and we're trying, this one wouldn't work for paternity, but we're trying to figure out who the dad is, how much of dad is going to match with son or daughter? About half, and then mom's gonna be the other. That's how you end it. Okay? Got
Do
it?
you need to have the moms figure this?
Well, no, because there's gonna be a large percentage from dad matching the other dads. Unless for some weird reason, the dads are related to each other and mom was with both, but that's weird. So, yeah.
That's not impossible.
It's not impossible, no, you're right. So, all right, I'm done.
Are we talking